Hello, and welcome to this introduction to the Brannigan Lab, the computational biophysics lab in CCIB. Why computational biophysics? Because biology can get really small and moves really fast. The reality is if you look at the top of this image, and if you actually look at that with an electron microscope, you end up getting a picture that looks like this. And this is about as good as it's going to get. What you need to realize is that this line that I've drawn over in red is actually a lipid bilayer that's got hundreds of protein complexes embedded in it. Because the interior and exterior of our cells are a very fluid dynamic environment, all of these proteins and indeed the membrane itself are waving around, reacting to things in their environment, and changing their shape or conformation at a rate that's truly unimaginable. And so you might say, well, I want to see what's happening in real time at a molecular level. And the answer is you can't. It's just not possible. And so while an experimental physicist may go off to try and design the next electron microscope, a computational physicist would say, well, you can't see what's happening, but you can predict with reasonable certainty what will happen by using some math, some Newtonian physics, and a lot of computers. So what we do is something called molecular dynamic simulations. We use it to simulate membrane proteins, especially ion channels. We simulate membrane lipid compositions and domain formation. So you see here is a nicotinic acetylcholine receptor, and it's surrounded by a pretty randomly mixed group of lipids and cholesterol. And as soon as we start the simulation, you can see that immediately things start to group together. It's not random anymore. There are very clear domains formed. And then on the right, we have an example of investigating some protein membrane interactions. So when you put a protein into a membrane that's a little bit short, what happens is immediately the membrane kind of adjusts and jumps up to accommodate that shorter protein. So why do we do these things? Well, the first project is looking at drug or other agent design. One project we do looks at the GABA A receptor and how it responds to common anesthetics that are used during surgeries. The center panel shows research that relates to frog eggs. Commonly, when people do wet lab research on the nicotinic acetylcholine receptor, they do so by putting that protein inside of frog eggs as their experimental medium. The problem is these researchers are using frogs' eggs to examine neuromuscular disorders in humans, and frog eggs have a very different lipid composition than human cells do. So what we want to be sure of is that this difference in lipid composition doesn't have any effect on the protein itself and its function. Finally, the panel on the right shows some coronavirus research that we are doing. The protein you see embedded in that membrane is the envelope protein of many different coronaviruses, including COVID-19. And this envelope protein is implicated in helping membranes to bend, which consequently helps the virus to escape from our endoplasmic reticulum and from our cells, further spreading the virus. In addition to using Amorel, Rutgers University's high-performance computing cluster, our lab has special access to Caliburn, the largest supercomputer in New Jersey, and pretty high up in the list of the world's top 500 supercomputers. If you choose to rotate in our lab, you may find yourself working on a protein like BDNF. BDNF is interesting because it is an intrinsically disordered protein. While we think of most proteins as having a set shape, this one does not. Even more concerning, however, is mutation of just one amino acid residue in this protein has been shown to increase the incidence of cancer. So from those of us in the Brannigan Lab, we hope to see you in rotation soon, and we can't wait to answer your questions. Thanks. Have a great day.